Okay, there it goes. Is that it?
Yep. Wait. Okay. All right. Okay. Welcome. So I'm going to start to talk about what happens from the point of where the DNA needs to be transcribed because the cell has decided, anthropomorphizing a bit, that protein is needed. So, all right. So here we go. All right. So we're going to start with DNA. And DNA is organized into large sections, and each of these sections have a lot of information in them. The sections that concern us are called genes. Right? And there are maybe 100,000 anywhere, from tens of thousands to 100,000 genes in higher organisms. Some people say we have hundreds of thousands of genes. It may well be that we have that many genes. These are large sections of DNA. They're large sections of DNA, and they have lots of different parts to them. Okay? There are parts that control the rest of that section of DNA. There are parts that let things bind to them. There are parts that don't seem to have any meaning at all. They're all encompassed in this one long thing called the gene. The part that interests us today is the part of the gene that codes for a polypeptide. So genes are long sections of DNA. So long sections of the nucleic acid, deoxyribonucleic acid, and these long sections of DNA contain in them the part that interests us, and those are the parts that code for polypeptides. They code for polypeptide. They contain clues, information that the cell will use to determine how to make a chain of amino acids. So by polypeptide, we mean a chain of amino acids.
The Lord of the Flies has a similar influence to that of the snake that influenced.
Right? And so the gene, among all the other all the other things it has, is sections that are saying this is how you're going to make amino acids. The DNA itself is a long sequence, as far as concerns us, is a long sequence of what we call bases. So there are long sequences of bases. So once again, we're talking about the DNA, in particular the gene section of the DNA, and inside that, there are long sequences of bases. Now, hopefully you're already familiar that there are 4 basic bases, and we symbolize those with just 4 letters. A, T, G, and C. So each of the bases corresponds to a letter, and these letters, or these bases, are repeated ad infinitum for a long time along the whole section of DNA. Okay? So for example, a section of DNA might have just a whole bunch of different letters after one another all along as we go. You're already aware, perhaps, that the DNA is double-stranded. In other words, DNA is actually 2 strands of DNA that come together to make what we call a double helix, a double-stranded DNA. And along the DNA, the bases are paired on each strand opposite each other according to the rules. All right, so you're already familiar, let's hope, you're already familiar with the fact that the DNA itself is 2 strands. So I've drawn 1 strand of DNA here. There's another strand of DNA right next to it most of the time, and it has base pairing. These are the 4 basic bases you have. We're symbolizing them with A, T, G, and C, and they pair among each other. The C is going to pair with G, the G is going to pair with C, the T is going to pair with A, and the A is going to pair with T. So if this is our 1 strand of DNA, right across from it is another strand of DNA, and it has complementary base pairing. So across from the A will be a T. Across from the T will be an A. Across from the G will be a C. Across from the C will be a G. And so on. Okay? So we've got two long strands of DNA. I've just drawn a little section of that strand of DNA here, and I've shown you how, as we go along the DNA, there are all these base pairs, and we symbolize these base pairs with these letters. Okay. The DNA, in fact, is made of two strands, so there's a strand with all these base pairs on one side, and on the other side, there's a strand with all these other base pairs. Okay, so we have two sets. And there's lots of base pairs. It goes on and on and on and on for hundreds of thousands of base pairs. And that's our DNA model. Okay? Sections of those base pairs are called genes, and within that is a section that codes for a polypeptide. So we're pretending that we're in the middle of the gene now, and we have this section, and this section is going to tell us something about a polypeptide. It's a code. It's not the polypeptide itself. It's not the chain of amino acids, but it's going to tell the cell how to get to the amino acids. The process that we meet first is called transcription.
So our symbolism
And in the process of transcription, we make a copy of the DNA. Now we already know that this DNA is a code for polypeptide, so that copy is also going to code for polypeptide because it's a copy of the DNA. And to understand transcription as much as we need, we have to understand that the DNA has a direction. The base pairs aren't read at random, it has a direction. And biologists, I guess, simplify the direction by talking about the carbon atoms, I guess, that are part of the molecule. And all we need to know from that is that the two directions are either from 3 to 5 or from 5 to 3. So I'm just going to assign directions to this little strip of DNA here. I'm going to say that this direction is 3, and we call it 3' for biochemical reasons that we don't need to go into. And then the other section here is called 5'. And the DNA is running anti-parallel. It's running in the opposite direction in the other strand. So this here will be the 3' end of this strand and the 5' end of its complementary strand. So every piece of DNA in its double strand has 2 strands, and I'm going to illustrate that with these meter sticks. So I've got this meter stick here, and it's running from 1 through to 100 in centimeters. The other meter stick is running, as I put it upside down to get it right, is running in the opposite direction. The numbers are still paired as we go along, but it's running in the opposite direction. That's what we mean by anti-parallel. Okay, and it's just a trick of the molecule. The molecule is built a certain way so that one runs in one direction and the other molecule runs in the other direction. And all that's important to us from that is that there's a directionness to the DNA. We put the top strand, we're going 3' to 5', and the bottom strand, we are making 5' to 3'. If we cut the strand in the middle, we still get the same direction. It's kind of like a magnet. You ever cut a magnet and you realize you cut a magnet, you still get a north end and a south end because it still has a directionality to it? It's the same thing for the DNA molecule. So let's take this top strand, which is 3' to 5'. If I only took a little section of the strand, it would still be 3' on this end and 5' on this end, because it has a directionality to it, really. If I cut it again, it still would be 3' over here and 5' over here. No matter how I cut it, that end is kind of always going to be the 3' end, and that end is the 5' end, because every single one of the base pairs is lined up 3' to 5' as you go along. Yes?
Is there ever like a point in the middle where you
What do you mean by point in the middle?
like the
No.
spot where
No.
5 pounds, 3 pounds?
No. No. Another good example is these board pins. See how these board pins stack together? This would be the 3' and this would be the 5'. Even if I break it, it's still got a 3' end and a 5' end. And the piece I broke off still has a 3' end and a 5' end. Okay, it's a really good analogy. So it doesn't matter if we just have doesn't matter how long it is. One end is still going to be 3' and the other is 5'. Even if we go to individual little pieces, we still have a 3' end and a 5' end. And the thing we know about DNA is that the other strand of DNA is running in the opposite direction. So I've decided that this is 3' and this is 5'. The other strand of DNA is running in the opposite direction. It's running 5' to 3', and they're pairing all along. Okay? So it doesn't matter if you break them apart, they're still anti-parallel. So there's no middle to it. You see that? Okay. All right. So this process of transcription is making a copy of the DNA on our way to making the polypeptide. So to transcribe something is to make a copy.
It's going to be due on Tuesday. Because Monday's a Thursday.
All right, so we're making a copy of the DNA, and that's the process of transcription. Making a copy of the DNA simply involves reading one of the strands. And we only have to have one of the strands read, since there's complementary base pairing. If I didn't have the second strand, I could know what it says anyway. So only one of the strands needs to be read. Okay? Now, the copy of the DNA when we're doing transcription isn't going to be a DNA molecule. It's a molecule that's very similar to DNA, but is not DNA. It's called RNA, ribonucleic acid. So the process of transcription, we make a copy of DNA, but the copy of DNA is now, it's done in RNA. And there's two really important differences that you need to know about RNA versus DNA. First off, DNA is deoxyribonucleic acid. So it's got a different name. It's double-stranded. And it makes up genes. So these are conserved. They're kind of kept inviolate. They're kept in the cell. There's some changes in them, but the cell tries to keep them forever and ever. So the genes are kept. The cell doesn't want to get rid of the genes. It tries to keep them. They're always there. It may make a copy of them. It doesn't get rid of them. It's conserved. Okay? Yes? Question?
Do you think is like the conservation of the genes different than something conservative?
Yes. We're only talking about the process of protein synthesis now. You're right. There is a thing called DNA replication. And it's semi-conservative because you keep half of the DNA when you do it, and you make a new half to match it. That's why it's semi-conservative. The process of transcription is completely conservative. The DNA is unchanged by the process of transcription. It's kept. We read it, and they put it right back away. It's like a really precious book that the cell opens up when it needs to read, and then shuts it up and puts it back away when it doesn't need to read it anymore. All right, so DNA is deoxyribonucleic acid. It's double-stranded, and it forms genes, and it's conserved in the cell. On the other hand, we have RNA, and it's ribonucleic acid.
Do you mean conserved as in kept in?
Yeah, it means not destroyed. It means preserved. It means like we conserve wildlife. We don't go kill them. We leave it alone. Right? We keep them. So that's what I mean by conserved. You can almost say like preserved, right? Except it's not just left alone.
Yeah.
We open it up and we use it, but we don't use it up. We put it right back when it's done. It's conserved. Okay?
Reusable?
Reusable is another good term. It's reusable thousands and thousands of times. Okay, ribonucleic acid. It's single-stranded with very few exceptions. In other words, DNA likes to be double-stranded. On its own, it will make double strands. It always finds a partner and double strands up. RNA is usually single-stranded. And it is not conserved. It's made as it's needed and then destroyed later. So that's your kind of basic difference between DNA and RNA. The cell wants to keep its DNA. It doesn't like to change it. It likes to conserve it. RNA, on the other hand, make it when you need it. If you don't need it anymore, chew it back up into its base pairs and let it go. And the other difference about DNA and RNA are what makes it up. It uses 4 base pairs, or 4 bases, sorry. And the bases we've already symbolized with A, T, G, and C. RNA has bases that are very similar to the DNA. There's a little difference. The sugar that's bound up with the DNA is deoxyribose, and the sugar that's bound up with the RNA is ribose. So the sugars are a little bit different, a little less oxygen on those ones, and that makes the whole difference in the molecule. That's not really important to us. What's important to us is that the symbols for the bases in RNA are different. So it uses 4 bases as well. And those 4 bases are A, U, G, and C. So you see 3 of the bases are the same, but in the DNA we get this base, the T, but in the RNA we get this base, the U, stands for uracil. So the bases that are used in RNA are really similar to the bases used in DNA, with that one difference that wherever we would have seen a T, we now see a U. So we saw earlier that wherever on the DNA molecule there was a T, it was paired with A, and the A is paired with T. If this was an RNA molecule, the A would pair with U, and the U would pair with A. Okay, so that's a little bit different if we're dealing with RNA. So, once again, we're talking about the process of transcription. We're making a copy of the DNA, but the copy of DNA is going to be an RNA molecule. It's not going to be another DNA molecule. So we're going to go back to our DNA molecule here. And just out of the blue, I gave you this section of DNA. 3' end reads A, T, G, etc. And the 5' end ends in G, etc. And what concerns us in the process of transcription is what happens to this sequence of DNA as we go. Notice there were 2 strands of DNA, but we only are interested in reading 1 strand. Okay?
Does it always copy from the 3' to 5' strand?
Yes.
So the other one will be 5' to 3'.
That's right.
Always.
So we had 2 strands of DNA. Up a minute ago I had a 2nd strand of DNA. And we had 1 strand which is 3' to 5'. And the RNA polymerase, the enzyme that makes the RNA, is going to read just this 1 strand. The information is mirrored in the other strand, but only 1 strand is read. So yes. Yes, I'll talk about sense strand in just a second. So the RNA polymerase, that's the enzyme that produces RNA, reads the DNA in the 3' to 5' direction. So that's why I'm only concentrating on this 1 strand of the DNA, because I'm only looking at the strand that goes 3' to 5'. The other strand runs in the other direction, but the RNA polymerase, this particular RNA polymerase, isn't interested in the other strand. It's only interested in the 3' to 5' strand. It's RNA polymerase 3, just to be technical, and it just works on DNA to make a particular kind of RNA, the kind we're making now. As the RNA polymerase moves along the DNA, it grabs bases in order to make a copy of the DNA, but the copy of the DNA is not an exact copy, it's a mirror copy. Because instead of putting down the bases as they are here, it puts the complement bases, the ones that match. Okay? So this strand of RNA, this strand of DNA goes A, T, G, G, C, T, A, C, G. The RNA that's made matches its complement. It's not an exact copy, it's like a mirror image with the matching bases. So we saw before that A matches T, so the A now will match with a U. I'm going to put the code here over on the side just to keep it straight. A pairs with T, G pairs with C. And that's it. If we have RNA, that T is replaced with uracil, so it's replaced with a U. So now U pairs with A. So for RNA here in red, we've got to put the U in. If we're dealing with DNA, it's going to be just the T. Okay? So now the RNA polymerase, this is this enzyme, begins to read the DNA. It reads at 3' to 5', and it puts the bases in place. So across from the A, it puts a U. Across from the T, it puts an A. Across from the G, it puts a C. Across from the C, it puts a G. Across from the T, it puts another A. Across from the A, it puts a U, and so on. Now, the base is of the RNA are complementary to those of the DNA, but they're going to be anti-parallel as well too. So the result is that this RNA molecule that's going to be made is going to have this be its 5' in, and this be its 3' in. Because just like the DNA, the RNA, when it's paired with DNA, is anti-parallel. It runs in the opposite direction. Yes?
The RNA adds the complementary nucleotides in the spot have the 3' direction?
That's correct. The RNA polymerase reads DNA 3' to 5', but produces the RNA 5' to 3'. And it's kind of important that you remember that. You got to remember the direction these things are operating in, because obviously the codes are different, read in different directions. Like for example, this starts A, T, G. In the other direction, it's C, it's G, T, A. So it reads differently in the opposite direction. So these are very directional, and it matters what direction they're in. Yes?
That RNA produces an mRNA, right?
Yes. So we're going to give this a special name now you're talking about. We're going to call this mRNA, or messenger RNA, because it's carrying the message. And what is the message it's carrying? Sequence of bases that specify a polypeptide, which is a string of amino acids. Any questions so far?
No.
Good. So, RNA polymerase has done its job. It's made a section of RNA. When it's done its job, this RNA is pulled off, and there's some RNA processing. That doesn't concern us too much at this stage. We know that sections of the RNA are pulled out. There are some introns that need to be pulled out, some exons put together, and some cap added at one end and a poly-A tail added at the other end. That's RNA processing. What we've been concerned so far is how we get a copy of the RNA from the DNA. Yes?
So there's a stop codon, right? And that's when it stops and breaks off, and then the poly-tailed add on later.
Not sure that there's a stop codon. There is a termination region of the DNA molecule that says stop transcribing here. That's what you mean.
For the mRNA.
Yes,
For the mRNA.
yes. The stop codon that we're going to talk about is a different one that's inside the messenger RNA, and that's a slightly different thing. There is a region that's called the termination. And we're just going to call it a terminator. In other words, it gets to a section of DNA, and for some reason, when the RNA polymerase gets to that section of DNA, it stops making a copy. It will not be exactly the end of the polypeptide. It makes a few extra. Yes?
That area would be the highly concentration of G's, C's, right?
Not necessarily. As far as I know, the A tail isn't coded for in the DNA. It just does. There's a section that's your terminator, and that means the gene comes to an end. The copying of the gene ends there. the ending of the copy of the polypeptide sequence occurs there.
On the DNA strand.
On the DNA strand. So this is information on the DNA strand. So on the DNA strand is a section that says start copying here. And then a little later on, in the 5' direction, there's a section that says stop copying here. Yeah, that's the start. Okay, and then there's a termination sequence that says stop. But that has nothing to do with the message that's in the messenger RNA. That's a message on the DNA, and it's a message for RNA polymerase. On the RNA, there will be other messages, but they're not for this enzyme. They're for other enzymes. So the messages for the RNA polymerase are on the DNA. The messenger RNA will have other messages, but they're not for RNA polymerase. They're for something else. Okay? Alrighty. So now we have this section of RNA, and it's being made and it's being processed. So we're going to talk just briefly about the processing. Alright, so the RNA polymerase So we've got a molecule, a macromolecule, because it's hundreds of atoms in it. And we're going to call it mRNA, and that stands for messenger RNAs, as we've been discussing.
We're getting a better
Right? So the mRNA molecule is a messenger RNA molecule, and we'll abbreviate that with mRNA. All right. The mRNA has to be processed. And the processing doesn't concern us too much as far as polypeptide, the polypeptide sequence. We do know that introns are removed, and that's a whole other story.
They're removing 2 IB products. They're removing 2 IB schools.
We know that a G cap is added at the 5' end, and we know that a poly A tail is added at the 3' end. In other words, a whole bunch of A bases are just added at the end, hundreds sometimes.
Copy this.
The most cohesive notes I've ever written. Like, not in terms of just like my writing.
So by poly A tail, we mean a whole bunch of A's are just added at the end. So what that would look like in this diagram is once this messenger RNA is made, at the 5' end, a G cap, we call it, would be stuck on. So this is still our 5' end, and a G cap is stuck on. And then at the 3' end, a poly A tail is added on, hundreds of bases, and they are just A after A after A.
Mr. Blenman.
And that's stuck on the end.
Isn't it, I don't know if this is right or not, but is the G-cap and poly A tail added before the introns are removed?
I'm not sure either. I'd have to look that up. I'll add the little note about that right here. There it is.
I think it's
Scrolling.
before.
Okay.
In the editing. I think it's
before.
editing.
Yes.
What is the purpose of the G-cap and the poly A tail?
Yeah. They have to do with RNA processing and with RNA protection and recognition and transport. So they're important for the RNA processing, the RNA protection, and the RNA recognition, and the RNA transport. Yes?
Do they serve the same purpose?
they're not exact. They don't do exactly the same thing, but they're both involved in those processes. If that's what you're asking.
So processing, protection, transport, recognition.
Processing,
Protection
transporting.
and recognition.
What was that though? Oh, so this was just 4.
Processing,
I think you just have a little song in the background.
Yes.
What does the poly A tail do after it's added to the
Sits there. Yeah. So it's got, as far as we know, maybe those 4 functions, and it's just added on the end of the molecule.
Can you talk about this functions?
What you need to know is that the GCAP and the poly A tail are probably important in RNA processing, recognition, transport,
Alright.
processing, recognition, transport, and
protection.
protection.
Thanks.
PPTR.
There are enzymes out there in that cell that want to chew up that RNA. There are places in the cell the RNA needs to get to in order to have the next step. And there are configurations that the RNA needs to be in for the next step to occur. And this processing helps it. Yes?
For next year's big game, you should make your big game around this and have like people that want to chew up the mRNA strands, and they have to get away from the
That'd be so cool.
enzymes.
That would be actually really cool.
Human digestive or
Like, yeah.
Make it like an episode of Magic School Bus.
Well, there used to be a show with like a white blood cells.
Yeah. Okay. So, now we've got our
It's a
messenger
smart idea.
RNA
Don't look at me like that.
after its processing. This process that we've had up to this point takes place in the nucleus. The RNA, messenger RNA, now moves out of the nucleus into the cytoplasm. So the RNA polymerase releases it from it's released from the DNA template. The DNA template's left alone, it's conserved. Right? Double strands are pulled apart, RNA polymerase has done its job. Those strands are conserved, put away for another time. If it's a gene the cell is really interested in, it may send the RNA polymerase through again to make another copy and another copy. But it doesn't change the DNA, it's left alone. It's the messenger RNA that moves from the nucleus to the cytoplasm. Right? So we're outside the nucleus and we're near the cytoplasm now. We're going to learn in a little bit that some of this messenger RNA will go right over to the endoplasmic reticulum. And we'll talk about why that may happen. But it's outside the nucleus now and it's in the cytoplasm. And this is sort of where we picked up with our discussions, because I started with the messenger RNA has been processed and now it's ready for translation. So this is where our earlier lessons start out. What I've just told you now is the part that involves the DNA, the part I said I would review. Since we're on talking about the story anyway, we might as well go ahead and talk a little bit about translation. The process is now called translation. So the first part of this process is transcription. We're now at translation. So we've got the RNA, it's out in the cytoplasm, and we're ready for translation. Okay. Translation happens in 3 steps. Initiation, elongation, and termination. And we talked about this already, so maybe we'll go just a little bit faster. In initiation, you need to remember that there is a start codon on the RNA. That start codon is
AUG.
AUG. So somewhere in the sequence of bases, the cell machinery runs along till it finds our start codon. And when it finds our start codon, that will initiate the process. Codon means sequences of 3 bases together. So 3 bases means a codon. So we've got 3 bases together. This is a codon. This particular AUG is our start, or initiation codon. And that starts this whole process going. What does initiation involve? It involves the ribosomal subunit binding to the messenger RNA. Which subunit binds?
The small subunit.
The small subunit binds to the messenger RNA. There's a region of the RNA that codes for that. This is another reason why we've got to distinguish between the stop and start codons, the start codon and the termination codon on the messenger RNA versus the start and termination codon on the DNA. Do you see that since there is a region of the ribosome ahead, in other words, on the 5' direction from the initiation codon, that the initiation codon for the messenger RNA being going to protein cannot be the initiation codon for making the messenger RNA, because there are regions ahead of that where stuff has to happen, including the ribosome's binding. So the ribosome binds to the messenger RNA strand ahead of the start codon. And I've given you a sheet on this, right? A little diagram. And the little diagram will show you that on your messenger RNA molecule, there's a region ahead of your start codon that's for the ribosome to bind. So you have a ribosomal binding region, and that's ahead of your start codon. Yes?
Is the ribosomal binding region the same base pair or bases every time? Or
Yes, yes, or very similar. It probably varies a little by organism and by phylum, but it's very similar. Messenger RNAs are recognized sort of across the whole world, not even the animal kingdom, across sort of the thing. You can take a messenger RNA from a very, very different creature, put it in a new creature, and there's still a ribosomal binding region to it. And the ribosomes know that that's where they need to go. Okay? So there's a region ahead of the start codon. So obviously the initiation of the DNA transcription has to happen ahead of your initiation codon, right? Because all that stuff has to happen, including a ribosomal binding region. And that's where the ribosome binds. The smaller subunit binds first. Then we know that the large subunit associates with something called the transfer RNA, we'll talk about in a minute, carrying your first amino acid and your large subunit. So your small subunit and your large subunit bind to it. And there's my wonderful diagram. I'm going to redo that. So here is my small subunit, sort of binding, and the messenger RNA is sitting in there. And there up top is my large subunit. These are sort of globular protein RNA complexes. And you know already that there are 3 sites in this ribosomal unit. Think of 3 clefts. Think of a lump of putty or clay, and you sort of push 3 fingers into a side and pull it out. So there's 3 little divots or clefts in it, and those are your 3 sites. The 3 sites have names. We call this the A, the P, and the E site. Okay? And they're going to be really useful when we talk about elongation. Okay? All right. So, how does it proceed? Well, once the initiation codon has been reached, a molecule binds to that codon, and a similar molecule binds to all the codons, and that molecule is called a tRNA. So a tRNA comes in, and we're going to talk about tRNA over here for a second. A tRNA is a particular type of molecule called a transfer RNA. And it's abbreviated tRNA. What a transfer RNA is, it's a special RNA molecule, so it has all the characteristics that RNA has. We were told it was single-stranded. We were told it's ribonucleic acid, not deoxyribonucleic acid. And we were told it has those same 4 bases, A, U, G, and C. But now this is a tRNA, a transfer RNA, a different kind of RNA molecule. Okay. This kind of molecule is needed for this next step. This transfer RNA molecule has bases all along it, few dozen base pairs all along it, and on the bottom here of this cloverleaf design, it will have particular base pairs as well that always end up, no matter which transfer RNA, how it folds, with 3 particular bases unique to that transfer RNA there at the bottom of that T shape. As we draw it in this direction, we'll call this the bottom. And we'll make this one complementary to our start codon. So the first one will be
U,
good.
A, C.
All right. So this is a transfer RNA molecule. It has dozens of base pairs. It's a fairly long molecule. And it'll have 3 of them that are sort of on this end of the cloverleaf design, and they're designed to match the codons of the messenger RNA. So the messenger RNA is divided up into 3 base codon units. The transfer RNA, in all of its base pairs, has 3 bases that make a codon, but these 3 aren't called a codon. They match the codon. So what are they called?
Anticodon.
They're called the anticodon. So this section of the transfer RNA molecule is called the anticodon. Now, there's a little bit of confusion between a codon and a messenger RNA and an anticodon. The codons are these sections of messenger RNA simply read 3 bases at a time. Nothing tells us where a codon starts or ends other than the AUG tells us where to start. They're not automatically divided up into 3s. The molecule just looks continuous to anything other than the cellular machinery that decides AUG must be where I start reading. Other than that, it's just base pairs. It doesn't look any different. There's no little gaps, no commas to tell it how to read. It just starts at the AUG, and now we're going to read from there. All right? The transfer RNAs, there are dozens of types of transfer RNAs, probably about 60. And you can see why if you do a little bit of math. And each of them has an anticodon at the bottom. Yes?
And I'm assuming they're called transfer RNAs because they transfer like amino
They transfer
acids?
what?
Amino acids?
Exactly correct. They transfer amino acids. That's what they call transfer RNAs. So the transfer RNA molecule is a single-stranded RNA molecule with a codon, and if it's ready to go, at this end, it's been charged with an amino acid. There are only 20 different amino acids. So if everything was super efficient, you would only need 20 different transfer RNAs, 1 for each amino acid. But because the code varies so much...
This is just a reminder for track and field. There's meeting in the small gym at lunch. That's track and field. There's a meeting in the small gym at lunch.
But because the code varies so much, it turns out that we have about 60 different messenger RNAs, transfer RNAs. And just think about the math. How many different bases could fit in the first position of a codon? How many different bases could this be? 4, right? It could be A, U, G, or C. How many possibilities are there for the second? 4 more, right? Regardless of what the first one is, you still have the possibility of A, U, G, and C for the second. How many for the third? 4 more. So you have 64 different possible codons. So even though there are only 20 amino acids that are used, we certainly have run out of time for codons. And we'll pick this up next day.
I'm not satisfied at the end of this. Let's see.
I think once you do it, it should be fun.
Did you stop?
No, I didn't.